Hello and welcome to PW NEET English YouTube channel. I'm Shalini Ma'am, your botany teacher, and in this lecture we are going to delve into the chapter cell cycle and cell division. Well, if not for this magical cycle, we would never be able to grow, our wounds would never heal, and reproduction honestly would become quite challenging. This is one of the most beautiful biological processes to ever exist, and when you look at a cell that's dividing under the microscope, it will almost seem like the cell is performing a tightly choreographed dance routine where each structure is doing its own thing, and ultimately it ends with a grand finale where either mitosis or meiosis will take place. Any biology enthusiast will thoroughly enjoy learning about this process. Not only is it interesting, it's also super important for your NEET exams. If you look at the data post-COVID, you will notice how every year a minimum of 4 questions have been appearing from this chapter. It is an easy chapter to understand, but remembering all of the events can be a little daunting. In order to make it easy for you, I will be sharing class notes along with some DPP questions to practice on the PW app. It is a free batch which is called Complete Botany by Shalini Ma'am. You may access this batch using the link given in the description box. Now, while you're on the app, you might as well explore all the other resources that are there, using which many students in the past have achieved their dream of becoming doctors. Now, without any further delay, let's get the session started. Alright, now, let's begin by talking about how our life began on this planet. Okay. If I ask you that, you'll say, oh, ma'am, our life started when we were born. Of course, on the day we were delivered into this planet, on the way we got out of our mother's womb is when we celebrate our birthday and all of that. Fair. But if you think about it as biology students, did life begin in that organism on the day it was delivered into this world? Not really. The moment the male gamete and the female gamete fused to give rise to the zygote, life had already begun. Right? So, not just in you and I, if you think of any sexually reproducing organism, it could be some other animal or some other plant. In all sexually reproducing organisms, life begins as a single cell itself. Today, we might be made up of trillions of cells, or a baby when it is born, it might have millions of cells at least. Regardless of how many cells we have today, when life started in us, it was with a single cell. And that single cell is the zygote. The zygote forms when the male gamete fuses with the female gamete. So, if we started our life as a single cell, how did we go on to become an organism with numerous cells? The answer is very simple, very obvious. It is by cell division, right? So, one cell that was formed, the zygote, it didn't remain as a single cell. It went on to undergo many cycles of cell division, where one cell became 2 cells, 2 became 4, 4 became 8, and so on. The number of cells kept on getting increased as the organism developed. Simultaneously, differentiation also happened. Specific cells took up specific functions in that organism, and eventually a fully functional little baby was formed, right? So, it's because of cell division that unicellular single-celled zygote gave rise to a multicellular organism like you and I. So, yeah, not just us, in all sexually reproducing organisms, we start our life from this single cell called the zygote. Now, talking of cells and cell division, if you consider certain characteristic features of cells, you can say that cells display 2 hallmark characteristic features. The first one is growth and the second one is reproduction. What do I mean when I say growth is a characteristic feature of a cell? I mean to say that cells increase in size. Growth is nothing but increase in mass of the cell, right? The cell, when it is formed, is very small, kutti baby cell. As it grows, it will accumulate more and more cytoplasm. The cytoplasmic volume will increase, more organelles will increase, and the cell will start appearing bigger, right? So that is growth inside a cell. Talking about reproduction. What do we mean by reproduction? Reproduction is a process by which living organisms give rise to young ones similar to itself. Like when I talk about reproduction in humans, whenever somebody is having babies, that is called reproduction, right? So you're producing another organism that is very similar to you. Apply the same context to cells. When we say cells are reproducing, what do we mean? There was one cell that gave rise to another cell which was very similar to itself. How does that happen? That happens by cell division. When an existing cell divides and gives rise to 2 cells, we say that reproduction has happened. The cell has reproduced. In fact, if you think about unicellular organisms like amoeba, okay, Let me quickly draw here an amoeba. This is one amoeba. When it was formed, it was very small. It grew in size. Growth happened, and after it grew to a considerable size, it went on to divide. When it divided, it formed two daughter amoebae. Now, when this cell division happened, from one cell we got two cells, and these two cells are similar to this cell. Initially we had only one cell, now we have an extra cell. That means reproduction has happened, right? So here in unicellular organisms, we can say growth and reproduction are synonymous. When we say unicellular organisms have grown, bacteria have grown, amoebae have grown, what do we essentially mean? Can a bacteria grow big enough to look as big as us? Can amoeba grow as big as the size of my palm? It cannot. When we say a unicellular organism has grown, we don't talk about growth in terms of its size. We talk about growth in terms of its cell number. So one cell was there, now it has become 50 cells. That means growth has happened. So when the number of cells increase, we consider that as growth. So over here, growth plus reproduction both can be used synonymously. So when cell division is happening, the cell number is increasing, growth is happening. When cell division is happening, newer organisms are added to the population. So reproduction is also happening. So in case of unicellular organisms, growth and reproduction can be considered synonymous, but that's not the case for us multicellular organisms. Growth and reproduction are mutually exclusive events in us. And even if you look at humans like us or any other organism, any other animal, when we talk about growth in us multicellular organisms, it can happen because of two reasons. It can happen because the number of cells in our body increased, or because the existing cells grew bigger in size. See, when a baby grows inside the womb, or a little baby after it is delivered grows into a toddler, obviously the size of that organism is increasing, but inside the body what is happening? The number of cells could be increasing. From a few million cells to many millions of cells or billion cells, the organism could have grown. So the number of cells inside the body is increasing. Right now, I am an adult, you are almost adults. So when I grow right now, it's not likely that I'm growing much because of the increase in the number of cells. If I'm growing, if I'm putting on weight or something, it is more likely because my fat cells in the adipose tissue, it's accumulating more fat and becoming bigger and bigger. So what is happening? The size of the cell is increasing. If in an organism there were 50 cells to start off with, and 50 cells divided to give rise to 100 cells, then that is also growth. If that organism has 50 cells, it did not undergo cell division, those 50 cells itself increased in size, that is also considered growth. Okay. So even in my body, growth can happen either because of increase in cell number or because of increase in cell size. Regardless of how growth is happening, it could be increase in number or it could be increase in cell size. Can I say just because I grew in size, I have reproduced? I can't say that, because reproduction is an entirely different event here. Growth is a different event. Unlike unicellular organisms and multicellular organisms, growth and reproduction are two entirely different, unrelated events. Okay, so they are mutually exclusive. Now I told you about these two characteristics of cells. The same characteristics living organisms also have. Even in living organisms, unicellular or multicellular, you most usually see growth and you also see living organisms reproducing. So growth and reproduction are characteristics of our individual cells as well as entire living organisms. Keep that in mind. Now this chapter is called cell cycle and cell division. Of course, in your lower grades, you would have learnt about cell division, the different types, mitosis, meiosis, but you may not have necessarily heard about the term cell cycle. What do we mean by cell cycle? You all know what a life cycle is. Think about our life cycle. Our life cycle begins the moment we are born, and then when we die, our life cycle ends, right? So within our life cycle, there are so many phases. When a baby is just born, it's a little cute baby, then you call it infancy. A little older kid, you call, what is it called? Toddler. It's called toddlerhood. After that, you call it a kid. Then people of your age, we call adolescents. People of my age, we are adults or middle-aged individuals. Then post our age, older people, aged individuals are there. So there are many phases in life cycle of us also. Each phase is represented by certain events, certain characteristic features. Like in infancy, the baby learns to swallow food. That is one event that happens. When it is an infant, it will learn to crawl. It will learn to stand up. It will learn to sit down. It will learn to walk. All of these are certain events that are associated with specific phases of our life. When it comes to your age group, your phase of life, you have certain characteristics like there's puberty happening, so you may start displaying secondary sexual characters at this point. So every phase is characterized by certain events. Like if you go to old age, their organs will start deteriorating, and their sense organs might not work as good as it used to. So those are some things that you see in older individuals. Similar to how our life has so many different phases, the life of a cell also has so many different phases. And each phase is characterized by certain events taking place. Now, our life starts with birth and ends with death. When does the life of a cell begin? When does it end? What are the phases? All of that we will cover under cell cycle. So, cell cycle is nothing but the life cycle of a cell. When does the cell's life begin? A cell's life begins when it is newly formed. Let's say there is one big cell. This is a pre-existing cell. This cell divided to give rise to two Botany baby cells. The life of this cell began the moment it was formed. Okay. Remember, cells cannot arise out of nowhere. If a new cell has to form, it can only form from a pre-existing cell. We've learned about that from the cell theory. Rudolf Virchow says, omnis cellula acellula. A cell cannot be formed out of hocus pocus magic. It can only form from a pre-existing cell. So when a cell has been formed brand new, that is when its life begins. Alright. So the life cycle of a cell, this is the starting point. What all happens through the life of a cell, I will tell you today. Okay. So I'll give you an overview of all the things that will go through, that this cell will go through during its life. And that itself is the cell cycle. All of the events that take place together is the cell cycle. So when this new cell is formed, it is very tiny in size. It is just born like a little baby, very small in size. Over the course of its life, this cell will start growing. When I say it grows, I mean to say it grows by increasing in size. Okay. So what was once a kutty small cell has now increased in size to become a much larger cell. How has this cell become this cell? The cytoplasmic content has increased. The volume of the cytoplasm has increased. The nuclear material, the DNA content has not changed. Okay, so it's just grown in size. What exactly do we mean when it accumulates more cytoplasm? When the volume of cell increases, basically more organelles are synthesized, more biomolecules are synthesized in the cell. Within a cell, a lot of metabolic reactions happen, right? So all of those metabolic reactions are actively happening, and the cell is accumulating a lot of organic material. Biomolecules are being synthesized, newer organelles are forming to accommodate make sure that the larger cell can function better. So newer organelles, newer chemicals, all of that will get synthesized, and as a result, the cytoplasmic volume will increase and the cell will become bigger in size. Now, this will further increase in size with time. How much can a single cell increase in size? When we talk about unicellular organisms, we have multiple examples. You have protozoans, you have many types of algae, you have bacteria. All of these are unicellular organisms. Did I say multicellular? I meant unicellular. So these are unicellular organisms. You don't really come across many unicellular organisms that are macroscopic, which you can see with your naked eye. All of these unicellular organisms that we talk about, like these bacteria, unicellular fungi, and these algae and all of that, protozoans and all of that, they don't really grow too much in size. Their entire organism is a single cell, but it does not grow beyond a point. You don't see any single cell as big as the size of this remote. You don't see any cell, of course, keeping aside the eggs. I know you'll come at me for that. Apart from that, you don't really see these organisms that are very clearly visible to your naked eye, right? They are mostly microscopic. Why is it that a single cell cannot grow as big as me? What is stopping it from growing and accumulating more and more cytoplasm? It cannot do that because if it does so, the cell cannot function normally. The cell can grow only up to an extent beyond which it will not be capable of growing, because if it does, then the cell will be at problem. What problem are we talking about? See here. From here to here to here, I've increased the size of the cell, the cytoplasmic volume has increased, but did you notice there is any change in the DNA content? There is no change in the DNA content. Whatever DNA was there here, 46 chromosomes, let's say, the same amount is there here and here also. We know that genetic material, DNA, this has information for pretty much everything that happens inside a cell. This is like the big boss sitting inside. Now, this has to control all of the activities that are taking place inside the cell. Now, the activities have become more, metabolic reactions have become more, but this guy, nucleus, the DNA content that is there, becomes incapable of controlling everything. You can think about it like this. Imagine there's a teacher in your class and there is a class of 50 students. The teacher can very well handle and manage a class of 50 students because everybody is visible to the teacher. Okay, the teacher will know if somebody is sleeping in the fourth bench, if somebody is scrolling reels on the second bench. It's easy. Also, the teacher can assess whether a student has understood a concept or not by looking at their facial expressions itself. So when there are 50 students under one teacher, there is personalized attention, there is better learning, teacher is happy, kids are happy, everything is going well, organization is happy. Imagine instead of 50 students in the same room, 100 students are crammed and you have only one teacher. Now it becomes quite tricky for the teacher to manage the class. Some students at the back could be sleeping, somebody could be singing, somebody could be listening to music, somebody could be, giggling in some part of the class, and the teacher can't really make out who is able to understand, who is not able to understand. The students aren't learning, the teacher isn't happy, the parents are complaining, and the organization is a mess. That is what happens even over here. The nucleus is given a job that it cannot take. It is too much for the nucleus to handle. Beyond that, the cell cannot grow. Okay. So basically when we say the cell has increased in size, cytoplasmic volume has increased, but the nuclear content remains the same, DNA content remains the same, we say that the nucleo-cytoplasmic ratio has been disturbed or disrupted, however you want to say it. So this is one reason why cells do not grow in size beyond a point. You do not find unicellular organisms that are huge. Okay, that's one reason. That's not the only reason. There's another reason why cells cannot function normally if this cytoplasmic volume is huge and there's only one nucleus. Because you see, for a cell to function, a lot of communication has to happen within the cell, in between cells also. Okay, so how does this communication happen? It happens by means of molecules present inside the cell. If a cell has to survive, it requires energy. How does it get energy? It gets energy from nutrients, it gets energy from oxygen. So basically it oxidizes the nutrients and obtains energy from that. So basically there's a lot of molecules like oxygen, nutrients, signaling molecules that need to be transported within the cell, and it mostly happens by diffusion. When there are too many molecules inside the cytoplasm, let's say there's one chemical here that needs to communicate something to another chemical here. If it has to get transported from here to here, the distance is too high in a cell, and diffusion is not a rapid process, right? And within the cell, it's not like one free swimming pool space. There are so many organelles in between, cytoskeletal elements between, so there's a lot of resistance. And despite all of that, if it is slowly moving by diffusion, it will take forever to reach wherever it's supposed to go. So transport of molecules will become quite a hassle. Oxygen, even if it's transported, will not get distributed inside the cell very well. Nutrients cannot get distributed inside the cell very well. Signaling becomes a problem because too much cytoplasm, too much volume, diffusion will take much longer. So on time, if a specific signal does not reach its target, the functioning will obviously falter, right? So that's what happens when the cell increases too much in size. To help you understand how this works, I'll give you a very simple analogy. What you're looking at this picture are 3 agar blocks. Okay, so these are agar blocks. You know what agar is? Agar is a sort of hydrocolloid that we obtain from marine algae, like red algae, that's used in your jellies and sometimes tarts, ice creams and all of that. So we have made 3 different agar blocks. Okay, so we've placed this agar block in a solution. Very rarely we can get to use black color here because the board itself is black. This is one such time we can write in black color. Okay, so within the solution, what have I placed these agar blocks in? I've placed them in a solution which is sodium hydroxide. As we know, sodium hydroxide is an alkaline solution. Along with sodium hydroxide, I will add a dye called bromophenol blue. This bromophenol blue is basically a pH indicator. Okay. Now that it is in contact with alkaline solution, it has taken up the blue color. Okay. Whenever it is in alkaline solution, it will appear blue. But if you have the same bromophenol blue in acidic solution, it will change color. Okay, so what I'm going to do right now is I'm going to move these agar blocks into a solution that contains vinegar, which is a very dilute acetic acid. Nonetheless, it is an acid. So once you move this agar block, these agar blocks from here to here, you can see that with time, the agar blocks are changing color. They are not entirely blue here. From the surface, you can see that as vinegar is diffusing into the agar blocks, they are changing their color from bright blue to light yellow. Why is that happening? Because from the surrounding acetic acid solution, slowly, little by little, it's diffusing into the agar block. Agar block will have many microscopic pores through which these acetic acid molecules will dissolve. Okay, I mean diffuse, not dissolve. It will diffuse. I want you to notice the size of these 3 different molecules. This one is, of course, the largest. This is the smallest. You can't even see there's a block that is existing here. If you can notice, the smallest block is the one that will get decolorized, become light yellow the quickest. The largest one will take a really long time. So after you've placed it in acetic acid solution, you pass it on. I mean, you wait for some more time and then you check again. You can see that at the same amount of time, this has almost become fully light yellow. This one is almost yellow with a little bit of blue in between, but this one has a really long way to go. Now, what does this experiment prove to us? It means that if the cell is smaller, diffusion of substances from outside can happen quicker. If the cell is very long, not very long, very large, the diffusion of substances from outside, it could be nutrients, it could be oxygen, whatever, from outside to inside to reach the deepest layers of the cell, deepest parts of the cell, it will take a much longer time. So, certain parts of the cells, if it is too large, will be deprived of nutrients and oxygen for a long period of time, which is not ideal for a cell. So that's why cells, being the clever things that they are, they know that if it grows too big, it's not going to work out in its favor. So it's like, okay, I'm done. This is all I can do. I've done my best. Now I can't handle it anymore. Let's divide and become smaller again. Okay. So once the nuclear cytoplasmic ratio has been disturbed, the cell will make a decision to undergo cell division. Okay. So this is one of the reasons why it decides to undergo cell division. Another reason is that diffusion will take a very long time. It could be diffusion of substances from the surrounding fluid or blood into the cell, or within the cell only molecules getting transported will take a hell lot of time, which is not ideal for a cell. So it will decide to undergo cell division. It can't just undergo cell division randomly like that. You know, within the cell, there is DNA, right? There is DNA here. In different segments of DNA, you have different information for different things. Here, there could be information for an enzyme required to break down proteins in the food you eat. Here, there could be information for an enzyme uh, that is involved in the formation of ATP in your cells. Here, there could be information for a pigment, an enzyme that produces a pigment responsible for the color of your eyes or your skin or your hair, whatever. In different regions, you have information for different things. If a cell decides to divide and randomly splits somewhere like that in the center, and this forms one cell, that forms one cell, this cell will not have information for these, this cell will not have information for this. So the cell that forms as a result will not have complete genetic information, and such cells will fail to function. Okay, so such cells are abnormal. For a cell to function normally, all of the genes are required, all the essential information is required. So you can't randomly cut apart DNA and say, okay, use this cell, take this, the second cell, this is for you. You can't split DNA like you split your ancestral property. So what does the cell do once it makes the decision to undergo cell division? I'll give you an example. Okay, right now think about it. Think that one of your seniors has made a beautiful notes out of NCERT biology, and that one fat book you divide into two parts. Exactly at the center there is one folding, and on one side, the first part of the book, there is all these notes, very beautifully written, handwritten mind maps and diagrams, and very engaging, and visual memory will be very great if you learn from that book. Such a kind of note somebody has made. Okay, so in the first half of the book there is information from all your 11th grade chapters, and in the second half of the book there is information from all your 12th grade chapters. Now you and your best friend, both of them want that book. Okay, so you go to your senior and he ask for that book. Obviously he's qualified NEET and he's in AIMS right now, AIMS Delhi. So you want that notes for your NEET preparation. So you go ask him for the book and he's like, dude, I have just one copy. I'll give it to you. You can make use of it however you like. Both of you are NEET aspirants. Both of you want to go to AIMS Delhi. Both of you want the complete information, both 11th grade and 12th grade. So what do? You don't just cut the book in the center and one person take first 11th grade syllabus and the other person takes 12th grade syllabus. That does not work, right? Because you're missing out on some information, the other person is also missing out on some information. So what would a wise person do in that case? If you are a very wise person, then you will take that book, go to a close by photocopy shop, get it xeroxed, make an extra copy. You keep one copy, you give the other copy to your friend, and you share the cost. Whatever was the cost, you can share that, split that between the two of you. That is a wise way to think about it, deal with it. The cells are super clever. They might come across as a very small structure inside our bodies, but they are super clever. So what they do is, they know that if they have to divide, both the cells that will form should get complete set of genetic information. So before they decide to divide, or once they decide to divide and before they undergo actual cell division and split, they will make an extra copy of its DNA. Okay, so whatever DNA is here, they will make an extra copy of that DNA such that both the daughter cells will get complete set of genetic information. Now this process is called DNA replication. Replica means exact copy. You look like your mother's replica means you're looking exactly like your mother. That's what it means. Replica means copy. So here DNA is getting replicated means DNA is making an exact same copy of itself, no change at all. Same thing, same information will be copied on to another molecule of DNA. So that happens. Okay, this happens when? Before the cell divides. So the cell grows, the cell replicates its DNA, and then only after DNA has replicated, the cell will undergo cell division and give rise to two cells, each with its own set of genetic information, complete set of genetic information. Now its life begins here. This will continue like this. Since this is a cyclic event, we call it the cell cycle. Now you might be wondering, why is it so important for DNA to get replicated? What about the other organelles? What about the mitochondria? What about the Golgi? What about the lysosomes? Does that also get equally distributed? Not really. It's not really necessary that that has to be equally distributed. If, for example, this parent cell had 20 mitochondria, it's possible, it's okay if one of them gets 11 and one of them gets 9. Depending upon its physiological activity, mitochondria can undergo fission and give rise to another one if it's necessary. If Golgi apparatus is less than 1, it can form newer Golgi cisternae. If lysosomes are less, Golgi can pinch out newer lysosomes. So depending upon the requirement of the cell, other organelles can form, but DNA is super precious because that is like the control center. There you'll have to be really careful and make sure that both the cells get the complete set of genetic information. Alright, so now, so these are some of the events that take place in a cell cycle. First, growth, then you have DNA replication, and then you have actual cell division. Though we call this chapter cell cycle and cell division, in a cell cycle, cell division is a very small part. In the cell cycle, growth and reproduction are also major events. So within the different phases of the cell cycle, when exactly does growth happen? When replication happens? When does the cell actually divide? What really happens when a cell is dividing? All of that we will learn in this chapter. Okay. Now, this was like an overview of what happens in the cell cycle. Now, let's take a look at the NCERT definition of the cell cycle. The cell cycle is defined as a sequence of events where the cell will duplicate its genome, which essentially means that DNA replication is happening. Also, it synthesizes the other constituents of the cell like other organelles and biomolecules, during which the cell will increase in volume and grow. And ultimately, it is going to divide and give rise to 2 daughter cells. All of these events together is called the cell cycle, and this happens in a highly coordinated, organized way. The cell cycle is so organized that it can put the most organized person that you know also to shame. So it is highly coordinated and it's under genetic control. It's not like cells can divide randomly, DNA can replicate randomly. A lot of enzymes control that. There are in fact a lot of checkpoints right within the cell cycle where it is checking for whether DNA replication has happened correctly, whether the chromosomes are correctly aligned at the center of the cell during cell division. So it's highly coordinated and under genetic control. Now I said that in the cell cycle, cell division is a small part. There are many other things that happens during the cell cycle. If you consider a human cell, for example, any human cell that is grown in laboratory conditions in a culture medium, human cells in culture, normally they divide once in every 24 hours, like once in a day, they will undergo cell division. And this is not normal, this is not a fixed number for every cell in our body. There are some cells that are replaced more frequently, there are cells that don't divide very often. This is like an average number. It's not like if one cell divided today at 5 p.m., the next day, exactly at 5 p.m., it's going to divide. So this is like a rough number. This duration or the frequency of the cell cycle can vary within our own cells also. Like in our body, our skin cells get replaced pretty often. The cells lining our alimentary canal get replaced pretty often, but the cells of our heart, the cardiac muscle, they don't divide very frequently. The neurons, they don't divide very frequently. So this is like an average number. So assuming on an average, the cell cycle in humans lasts 24 hours, and once in 24 hours if our cells are dividing, then more than 95% of the time of the duration, the cell is not really dividing, it is growing and doing other activities. The cell division will actually take a very short period of time. Now, in our cells only, every cell will not divide once in 24 hours. Within us, from one cell type to another cell type, it will vary, like I said earlier. The duration of cell cycle, frequency of cell cycle will vary from one species to another species as well. In yeast, which is a unicellular fungus, you can see that cells divide every once in 1.5 hours or 90 minutes. So how frequently the cell divides depends on the cell type, different cell types within the same organism, and it also differs from species to species. Okay. So the duration of the cell cycle is variable. Variable means it can change, it can vary. It varies from one organism to another. It varies from one cell type to another cell type. So typically in yeast, every 90 minutes, the cell will undergo cell division. Talking about what the cell does when it is not actually dividing, it exists in a phase called the interphase. In a cell cycle, you can find two phases typically. So here, let's say the cell cycle has started here. This is a kutti baby cell that has just now formed. In this clockwise direction, it is growing and the cell cycle is proceeding, let's say. From the time it is formed to the time it undergoes cell division, it exists in a phase called interphase. Inter means what? Inter means in between. You may have heard of inter-school competitions, inter-class competition, inter-house competition and all of that, right? Inter means in between. Why are we calling this interphase? The reason we're calling this interphase is because this phase is happening between two successive cell divisions. Just now one cell division has happened and a brand new cell has formed. So this cell is growing and it decides to divide here. Here is the next round of cell division. So it has just now finished a division, it's going to divide again. The phase in the cell cycle during which it has just now finished division and is going to undergo another division. The phase, the period between two successive divisions, in between, inter, that is called interphase. Okay. So the cell occupies most of its time in interphase itself. So here, let me show you. From the time it is formed to the time it decides to divide or undergo cell division, begins to divide, it is in a phase called interphase. Whatever I have outlined in pink right here, that is the interphase. The phase in the cell cycle when the cell is actually dividing is called the division phase or M phase. So that I will highlight here in maybe cyan color. So here is when the cell is undergoing cell division. So this is the M phase. From here to here, this is interphase. Okay. Now, like I said, if a cell is dividing once in every 24 hours, most of the time it spends in interphase and a very short period of time, it will undergo cell division. In 24 hours, cell division will actually take less than 1 hour or around an hour roughly. More than 95% of the time, the cell is spending in interphase itself. Now, within interphase, you have three subphases. I told you that this is interphase, right? Whatever I'm outlining in pink right now, that is our interphase. Right, within interphase, you have three subphases that are called G1, S and G2. First G1 phase, then S phase, then G2 phase, exactly in this order. Now, what really happens during interphase? We also call it the resting phase. But the cell isn't really lazy and resting in this phase. It has just now undergone cell division, so it's not immediately going to undergo another round of cell division. So between two rounds of cell division, it's resting, meaning it's not actively dividing anymore. That's what it means. So it's called the resting phase. During the resting phase, the cell is metabolically very active. It is performing a lot of synthetic activities. It's synthesizing a lot of biomolecules. Many new organelles are forming. It's increasing in volume. Basically, during the interphase, the cell is growing and preparing to undergo cell division in future. Okay. So during interphase, you can see growth happening and also DNA replication happening. Like I said, the interphase is divided into G1, S and G2. What does G here mean? G means gap. Okay. G1 phase is the gap 1 phase. Why is S called S phase? Because some very important molecules get synthesized in that phase. G2 phase is gap 2 phase. If you see here, here is where mitosis is taking place. The cell cycle is proceeding in this direction, clockwise. Okay. So G2 phase is a phase in the cell cycle prior to mitosis. Just before mitosis, you have G2 phase. Just after mitosis, you have G1 phase. That's why G2 phase is also called pre-mitotic phase. Pre means before. Just before mitosis, you have G2. Just after mitosis, you have G1. So this is called post-mitotic gap phase. This is pre-mitotic gap phase. And G1 is post-mitotic gap phase. Okay. So in your multiple choice questions, if they've given pre-mitotic and post-mitotic and not mentioned G1 or G2, you know what is G1 and what is G2. Keep that in mind. Now let's specifically focus on the G1 phase. I'm going to outline the G1 phase over here. As you can see, G1 phase immediately follows the M phase, and after G1 phase you have the S phase. S phase is called synthesis phase because here is where DNA replication happens. DNA makes an extra copy in the S phase. So if you see, G1 phase is like an interval between M phase, during which mitosis is taking place, and S phase, during which DNA replication gets initiated. Even though G1 phase is called resting phase or a gap phase, it's not metabolically inactive. A lot of metabolic synthetic reactions will keep taking place. A lot of new molecules and organelles will keep getting synthesized in the cell, because of which the cytoplasmic volume is going to increase, and as a result, the cell is going to show growth in this phase. But here is not where replication happens. Replication specifically happens in the S phase itself. Now speaking of, just a moment, I got the same slide again. Yeah. Speaking of S phase is between two gap phases, G1 and G2. This is a very important phase in the cell cycle because it is in the S phase or the synthesis phase that replication of DNA or DNA synthesis takes place. Once replication of DNA takes place, DNA makes an extra copy of itself. The genetic material, the content of DNA kind of doubles. When before replication, each chromosome in the cell will have one chromatid, but during replication, each chromosome makes an extra chromatid. It copies the information on one chromatid onto another chromatid, and chromosomes will have two chromatids. This is called 2C condition and this is called 4C condition. More about it I'll tell you. Okay. So the initial amount of DNA before it starts replicating is represented as 2C. Whatever content, additional duplicate content forms in a cell during S phase, that is called 4C condition, twice the amount of DNA. Now, if you see this and this, the number of chromosomes haven't changed. The DNA content has doubled, that's all. If you are counting chromosomes, we don't count chromosome based on how many chromatids are present. When you're counting chromosomes, we essentially look at the number of centromeres present. Over here you have 4 centromeres, here also you have 4 centromeres. So the number of chromosomes has not changed at all. It has been 4 here, it is 4 here as well. But what has changed is the amount of DNA because each chromatid has made an additional copy. The DNA content is doubled, but the chromosomes have themselves not increased much in number. So there is no increase in the chromosome number. In animal cells, the centriole duplicates in the cytoplasm in the S phase. Along with DNA synthesis, the centrioles also duplicate. In the previous chapter, we learned about what centrioles are. Centrioles are these hollow cylindrical structures that have a cartwheel-like arrangement of microtubules. They are perpendicular to each other. They are like the letter L and they are usually present towards the nucleus. Normally, animal cells have one centrosome per cell very close to the nucleus. All of that we've discussed in the chapter Cell, the Unit of Life. We're talking about the exact same centrioles. In this chapter, we learn about how exactly they help in organization of the microtubules, how they help in spindle formation and all of that. Okay? Yeah. So, here's how the centrosomes replicate themselves, make an extra copy of themselves. Let's start off from this point. There's only one centrosome with two centrioles perpendicular to each other with some pericentriolar material. First, the two of them are present, only 2 of them are present, eventually what will happen is each one will start producing centriole perpendicular to itself, and eventually you will end up getting 2 centrosomes inside the cell. Okay, so very close to the nucleus, the centriole will duplicate, the centrosome will duplicate. Centrosome is this structure with the 2 centrioles and the pericentriolar material. So we start off with 1 centrosome, but after the centrosome cycle, we will get 2 centrosomes. Okay. Now, why are 2 centrosomes essential? Because we are going to form 2 new cells. Each cell will need 1 centrosome. So an additional copy of the centrosome is also made. That's about S phase. And remember, centrosome duplication happens only in animal cells. Plant cells don't have centrosomes. Now, let's move on to G2 phase, which is our pre-mitotic phase right here. See, immediately after G2 phase, mitosis is going to take place, and for mitosis to take place, a lot of proteins are required and a lot of enzymes are required. So whatever proteins and enzymes are required for mitosis, all of them will start getting synthesized in the G2 phase in preparation for mitosis. Also, the cell continues to grow in the G2 phase. So proteins are synthesized and cell growth continues. I told you in cell cycle and cell division, in the cell cycle, 3 events are there: cell growth, DNA replication, and cell division. Now, cell division happens specifically in the M phase. DNA replication happens specifically in the S phase, but the cell continues to grow through G1, S and G2 phases. It's a continuous process, okay? But replication and cell division happens specifically in their respective phases. Now, here is a table showing what are the different events that happen during the phases, subphases of interphase. In gap 1 phase, the cell is growing, preparing for DNA replication that's going to happen. It's going to synthesize a lot of the enzymes and proteins that are required for DNA replication. During S phase, DNA replication happens, and if it's animal cells, the centrosome also duplicates. Histone proteins are synthesized. In G2 phase, it's the final preparatory phase for mitosis. Cell growth happens considerably, but along with that, the proteins that are essential for cell division, they get synthesized. Yeah. Now, there is one other phase called G0 phase, about which I have not mentioned here so far. This is called the G0 phase. It's shown here in your NCERT textbooks. So what exactly is this G0 phase? Let's understand that. You know, I mentioned how every one of our cells don't divide at the same frequency. There are some cells that are dividing very frequently. There are some cells that divide once in a while. There are some cells that don't appear to divide at all. For example, some of the neurons in our brain, they don't divide at all. Newer connections are formed between existing neurons, but neurons rarely ever divide. Same goes for cardiac muscle cells in your heart. They very, very rarely divide. They probably don't divide at all. So, if a cell is not dividing, which phase is it present in? If it's a constantly dividing cell, then it will keep going through these phases repeatedly in the cycle. But if a cell has not divided in a long time, it has stopped undergoing cell division, where exactly is it? Is it in G1, S, G2? Where exactly is it? It can't be in S or it can't be in G2. What basically happens in such cells is once it gets differentiated it grows. After it is formed, it grows considerably in size, but doesn't grow so much that the nuclear cytoplasmic ratio is disturbed. It will show considerable amount of growth, increase in size, increase in volume, and after that we say that it exits this cycle. It no longer goes on in this cycle. Once it has exited the cell cycle, it remains in a phase that we call G0 phase or quiescent stage, which means a dormant stage. Okay. It can exit the cell cycle only at the G1 phase before cell division or replication is initiated. So from the G1 phase, it exits the cell cycle. It is remaining in a sort of dormant phase called the G0 phase. If at all, that cell has been called upon by the body to undergo cell division. Now the body has some need and it requires that particular cell to divide so that the organism survives or something, let's say. Due to some condition, the organism requires that cell to undergo cell division. Under such cases, it re-enters the cell cycle at the same point that it left. It exited from G1, it will come back to G1. And only after it enters G1, it will produce the proteins and enzymes required for replication, go through S phase, replicate its DNA, go through G2 phase, produce the proteins that are required for successful mitotic division, and only then will it divide. So this phase is a phase where cells are existing when they're not actively dividing. Okay. So some cells in adult animals do not appear to exhibit cell division. For example, like I mentioned, some of our heart cells. Many other cells divide only occasionally as needed to replace cells that have been lost because of injury or cell death. So what happens to such cells? Such cells exit the G1 phase and enters an inactive stage called the quiescent stage or G0 phase of the cell cycle. Here I am saying it is inactive stage. Inactive is with respect to cell division. It's not actively dividing. That is what it means. That does not mean the cell is not doing any job and is being lazy and just sitting around and whiling time there. The cell will be actively metabolizing. When you say a heart muscle does not divide very often, does that mean the heart muscle cell, the cardiac muscle cell is resting doing nothing? Not really. If it rests, we'll all be gone, right? So they are doing their everyday activities, they're performing their metabolic activities. It has just been inactive with respect to cell division. It's not actively dividing. That's not, that's what it means. Okay. So cells in this stage remain metabolically active, but they no longer proliferate. Proliferate means they're not increasing in number, they're not undergoing cell division actively. So depending upon the requirement, they can anytime get back into the cell cycle, enter the cell cycle at G1 phase and continue to divide. Okay, so that's about our interphase. Now let's talk about division phase. During M phase, either mitosis can happen or meiosis can happen. First, we learn about mitosis. Later, I will tell you about how exactly meiosis goes on. Okay. So, M phase in your textbook is also called the mitotic phase. Mitosis is called equational division. I'll tell you why. Because when mitosis happens, one cell divides to give rise to two cells. One mother cell or parent cell gives rise to two cells. If the parent cell is diploid, the daughter cells are going to be diploid too. If the parent cell is haploid, the daughter cells are going to be haploid too. What does that mean? The number of chromosomes in the daughter cells is going to be equal to the number of chromosomes present in the parent cell. Now, why is this important? We are calling this equational division because in every type of cell division, this is not the case. In meiosis, another type of cell division, the number of chromosomes get reduced to half when cell division happens. Okay, meiosis happens, it gets reduced to half. There you call it reductional division. Here we call it equational division because if the parent cell has 46 chromosomes, the daughter cells are going to end up getting 46. If the parent cell has, let's say, 90 chromosomes, the daughter cells are going to have 90 chromosomes. There's no reduction, addition, nothing is happening. It's equal, exactly the same. That's why you call it equational division. Now, mitosis happens in plants and it happens in animals as well. In animals, mitosis occurs in diploid somatic cells usually, okay? But normally you don't see mitosis happening in the germ cells. Cells that are supposed to produce gametes will undergo another type of cell division called meiosis. Mitosis is usually in the body cells or the somatic cells. But in honeybee males, the males are haploid. All they have is one set of chromosomes. So in males, haploid cells undergo mitosis. Usually in animals, it is the diploid cells that will undergo mitosis. But in honeybee males, even the haploid cells undergo mitosis. Talking of plants, both haploid and diploid cells undergo mitosis. You've all learnt about the alternation of generation in plants, right? So you know how haploid gametophytic phase is there, diploid sporophytic phase is there. The gametophyte also, mitotically, it will divide and give rise to a multicellular gametophyte maybe. Even in the sporophytic phase, the diploid cells will undergo mitosis to increase in size and grow. So mitosis happens both in haploid and diploid cells in plants, but in animals, it's mostly in diploid cells, very rarely exceptional cases where it happens in haploid cells. Now, mitosis or the M phase is considered to be the most dramatic period of the cell cycle. There are some animations available for free on YouTube. You must watch it sometime. It's amazing what happens during mitosis. It's beautiful how the DNA changes its form, becomes chromosomes, aligns at the center. It's almost like a dance, routine is being performed, and it's so beautiful, so coordinated. And yeah, you should watch it. And I think in your schools also, in practical labs, they would have shown you how to, make slide preparations of onion root tips, where you can see different phases of the cell cycle, right? It's very, very beautiful. You should watch it if you haven't yet. So yeah. So during the M phase, major reorganization happens within the cell. More about it I will tell you. And in M phase, there are two subphases, karyokinesis and cytokinesis. Karyon means nucleus. Nucleo, I wrote nucleus. Cyto here refers to cytoplasm. So what is karyokinesis? It is the division of the nucleus. Cytokinesis is the division of the cytoplasm. When I say cell division happens, in the S phase itself, the cell would have made two copies of DNA. There are two nuclei present. Okay, so there's two set of genetic information present. First, what happens is these two nuclei will get separated. The DNA content gets separated. Post that, the cytoplasmic content will get separated because separation of DNA and distribution of DNA equally between two daughter cells is a very, very important task, because that's where all of the information is present. You can't mess up there. So that happens in a highly coordinated manner. Post that, cytoplasm splitting is a walk in the park, right? So that will happen very easily, smoothly. So even over here, we have to first start learning about karyokinesis, and then we'll proceed to cytokinesis. Karyokinesis further has four subphases, which is prophase, metaphase, anaphase, telophase. Okay, so here you have prophase, metaphase, anaphase, telophase. This entirely is your M phase. Okay, so this entirely is your M phase. Within M phase, you have karyokinesis, where the nucleus divides, and then you have cytokinesis. Now within karyokinesis, you have prophase, metaphase, anaphase, telophase. Exactly in this order it will proceed. Okay, so the correct order of these different phases you will have to remember: prophase, metaphase, anaphase, telophase. PMAT. Okay. Now, prophase is the first stage of karyokinesis. How do we know that the cell has entered prophase? There is a distinct feature, a characteristic feature, which marks that the cell has entered prophase. That is, DNA that has been replicated will start getting condensed. Condensation of DNA will begin in the prophase itself. What do I mean when I say it's going to get condensed? See, depending upon which phase the cell cycle the cell is in, which phase of the cell cycle the cell is in, DNA will exist in all phases, but it will not be in the same form. It will perform so many different avatars. Okay, so it will have different forms. Like normally when the cell is not dividing and the cell is in interphase, it will exist in this form, which is called chromatin. It's like long threads that are dispersed within the nucleus. If this is a cell, this is the nucleus. Within the nucleus, it is dispersed in the form of many thread-like structures like this that are called chromatin fibers. This is when the cell is in interphase. Okay. Now, does that mean the DNA is like this? No, DNA is very rarely like naked DNA. Always it will be associated with some proteins, and it is coiled to an extent. It's not like pure naked DNA ever. So, when the cell is in interphase, it exists in the form of chromatin fibers, not even like this. It's okay, maybe like this sort of chromatin fiber will exist. Now, think about it. If you have like a long thread, okay, so here you have a long thread, pink in color, interspersed with a long blue thread like this. If you want to separate the two out properly without messing up, what is one thing that you can think of? I would say we should start taking like a cardboard roll of sorts and winding this very slowly around this. When it becomes more compact, it is easier to separate these two strands of thread, right? What do you think? What do you think, people, ladies and gentlemen? So you take this thread and you start winding it around this piece of cardboard, and you take this thread and you start winding it around this piece of cardboard. So what's happening here? What was long and thin has become shorter, shorter, shorter. What was super thin has now become wider. Something very similar happens in the DNA also when the cell is undergoing cell division. So if DNA exists in the form of long, long threads interspersed randomly inside the nucleus, it becomes quite difficult to segregate them between two daughter cells. In order to make segregation easier, in order to make sure that both the daughter cells get complete set of genetic information, they undergo condensation, during which they will wind around proteins and become more compact structures. Now this is also DNA, here also there is DNA, here also there is DNA, chromosome also has DNA, but this is naked DNA, it's not associated with any other molecule. In all other condensed forms, it is associated to an extent with certain kinds of proteins, okay. So once it winds around proteins, then we say that it is condensed. Here, similar to how we use a cardboard roll to roll the thread around, we use proteins here to wind around the DNA, okay. Yeah. So now, this condensation will not happen as a single step, it happens as multiple steps. For DNA to go from this to this, it will take time. So it starts to get condensed in prophase, at the beginning of prophase, which is called early prophase. When that condensation has begun, we kind of get to know that, okay, DNA has started to condense, now it is in prophase, okay. So it's marked by the initiation. Why did a heart come instead of a star? Oh my god, okay. Initiation of condensation of chromosomal material to form compact pre-mitotic chromosomes. In the S and G2 phases, the new DNA molecules formed, they are not distinct. Like I said, they are intertwined. They look like these sort of threads that are interspersed. But during early prophase, they start to get untangled and become more and more condensed. Okay. So at this point, if you look at the chromosomes, you can see how the chromosomes will start appearing to have two distinct chromatids. Why do they have two distinct chromatids right now? Because prophase is happening only after S phase. During S phase, an extra copy was made, right? So each chromosome will at this point have two chromatids that are attached with to each other at the centromere. Now, by this point, if it is an animal cell, the centrosome also would have gotten duplicated. So here is one centrosome, here is the other centrosome with its pericentriolar material. It has gotten duplicated. Now this will start moving towards the opposite poles. It will start moving towards the opposite poles during early prophase. Late prophase, towards the end of prophase, almost transitioning into metaphase, you can see that the centrosome would have already reached the opposite poles. It started moving here. It has fully reached the poles towards the end of prophase. This again is only in animal cells, not in plant cells. You know how the centrosome is made up of centrioles, which are made up of microtubules. So these centrosomes will start radiating out microtubules. Radiating means if this is the centrosome, from here microtubules will start radiating. You know how radiation, sun's radiation, you draw rays like that, right? So from here, it will start radiating microtubules. Microtubules are very, very small tubes, like I told you in the previous lecture, cell the unit of life. So it will start radiating microtubules. When it starts radiating microtubules like that, it will appear like a star. Aster is another name for star. Okay. So now that it forms these radiating microtubules are being radiated, it appears like a star, due to which we call it asters. Again, this is only in animal cells. Apart from centrosome, do you observe any change? By looking at the late prophase diagram and the early prophase diagram, I want you to notice on the chromosome and on the nucleus, obviously, because that's the only other thing that you can see. You can see how the chromosomes condensation has started here, which is almost complete here. If you look at the DNA, here it is more thread-like, less compact. Here it has become thicker and shorter. And take a look at what has happened to the nuclear membrane. Here it is more or less intact, but here if you see, the nuclear membrane has kind of disintegrated. So by the end of prophase, during late prophase, you can see how the cells will not show any Golgi apparatus, endoplasmic reticulum, nucleolus, nuclear envelope, none of that will be visible. And it's almost like the chromosomes are lying freely in the cytoplasm. And you can also see faintly spindle fiber will start arising from either end of the cell, from either poles. That's the story of late prophase. Now I said the nuclear membrane, nucleolus, Golgi complex and endoplasmic reticulum, all of that will not appear inside the cell when you look at it when it's at the end of prophase. Now when I say it disappears, you no longer see it. What do I mean? Where have they gone? Where have they disappeared into? Let's understand that. This is something that most textbooks won't tell you. This is a cell cycle, and let me zoom it a little bit so you can see it better. Oh my god, this is not how a zoom is supposed to work. Let me try some other way. This around. So let's see. Turns out I can't. I don't know. I don't want to waste time figuring out how to do this. Oh my god, it's doing one discordance. So here's the cell in interphase. You can see how during interphase you have all of these organelles spread out inside the cell, right outside the nuclear membrane. You can see the endoplasmic reticulum. You can see mitochondria around. You can see a lot of vacuoles, lysosomes. You can see Golgi apparatus. All of that is visible. So what happens during prophases? Endoplasmic reticulum is nothing but membranous structures. Golgi is nothing but membranous structures. Nuclear membrane is membrane again. So all of these, when I say they disappear, I mean to say they don't just vanish out of the cell. They don't look like endoplasmic reticulum anymore. Okay. So all of these many network of sheet-like structures that you have spread out inside the cell, that is endoplasmic reticulum and Golgi apparatus, they will no longer have that endoplasmic reticulum like appearance, Golgi like appearance. All of those vesicles will get kind of detached and become thinner and smaller and go align themselves. You can see this is the transition phase during pro-metaphase. You can see how the nuclear membrane has started to kind of disintegrate here in prophase. This is the transition phase between prophase and metaphase, and you can see how whatever existed in the center is kind of moving towards the side. Mitochondria were spread out, they are kind of moving towards the side. If you come towards metaphase, you can see how they are almost all the vesicles, mitochondria, other organelles are spread out towards the outer surface, towards the periphery of the cell. So you no longer see it in its original location. It's no longer located near the nucleus. It no longer has its individual sheet-like structure. So it's become smaller, thinner, and it has moved to the periphery of the cell. That's what we mean when we say the membranes of ER and Golgi have disappeared. The nuclear membrane also forms little vesicles within which important nuclear proteins are preserved, and it also forms vesicles and moves and occupies the outer peripheral area of the cell. Okay, so why is that important? Think about it. When you're practicing dance for your whatever school day, annual day, whatever in school, sometimes I don't know if they still do it, but back when I was a kid, they used to make us practice in class. You're making way by pushing benches and everything towards the walls and make some additional space in the class so that people can practice there. Something similar happens here. It's not like they're vanishing, they're there, but they don't have their earlier appearance. They have broken down into smaller parts and they have occupied the periphery. Now, so much space is present in the cytoplasm in the center. This gives chromosomes sufficient space to align themselves. It gives the cells an opportunity to make sure that these chromosomes get uniformly divided between the two daughter cells. Okay. So that's what happens when we say that the nuclear membrane, the endoplasmic reticulum, Golgi disappear. What about nucleolus then? Nucleolus is a region inside the DNA where very actively ribosomal RNA synthesis will be happening. So different chromosomes have different regions where there is information for rRNA synthesis. For example, this is one chromosome, let's say. Okay. So let's say this is one chromosome. I'm not drawing it like chromosome, but let's say that's one chromosome which is in its chromatin form. This is one chromosome. This is one chromosome. Let's say this is one chromosome. In these chromosomes, exactly at this location, there are genes for rRNA synthesis. Wherever ribosomal RNA synthesis genes are there, all those regions will come together. In that region, they are constantly transcribing and producing rRNA, ribosomal RNA, and a couple of proteins also. So that region is called nucleolus. That is what appears as a dark spot inside the nucleus. But what happened during prophase? This thread-like DNA that was there initially is no longer thread-like. So what is happening here? So over here, this started to get condensed, right? What was initially like this thread became this. What was initially like a thread, the pink one became like this. Obviously, it's not going to be colored pink and yellow and inside the cell. Just for your reference, I'm showing this. Green and blue. Okay, so it becomes shorter and shorter. Now that it becomes shorter and shorter, it's drawn away from each other and you no longer see that particular area inside the nucleus. That is what we say when we say that is what we mean when we say the nucleolus has disappeared. It's not like there was some structure and abracadabra, poof, it is gone, disappeared. It's just that this region does not exist anymore because the DNA has condensed. They are no longer together over there. All right. Yeah. So that's about what happens during prophase. How do we know it is metaphase? We know it is metaphase when the nucleus is completely disintegrated. Okay, so it is marked by the complete disintegration of the nuclear envelope. In this phase, since there is no nuclear envelope, all of the DNA, which is now in the form of chromosomes, will come and align at the center of the cell, like somebody has asked them to form a line in the assembly. All of them will come and align at the center. Okay, another thing that I want to mention to you all, it's not like in the cytoplasm, these chromosomes are swimming freely and they go and join there. They're literally pushed. Okay, there are many motor proteins inside the cell that literally push these chromosomes and make sure they go and align themselves at the center. In this process, a lot of microtubules, a lot of motor proteins are involved. So it's not like the chromosomes already know, they just have to go and align themselves there. So it takes effort and motor proteins do the job. It's not like floating, they come and stand in the center. Okay. So all the chromosomes are spread through the cytoplasm once the nuclear membrane disintegrates. But during metaphase, they will all come and align at the center of the cell. Yeah. So this is called the metaphase plate or the equatorial plate. At this point in the cell cycle, you can see the chromosome structure very, very distinctly. It is in their highly condensed, highly compact stage in metaphase. And in fact, if people have to study the number of chromosomes in an organism, the structure of chromosomes, do like prepare a karyotype of any organism or any type of cell, then this is the best time to do it because they are most condensed and can be very, very easily observed under the microscope. They look beautiful, beyond beautiful. So yeah, that's metaphase. Now, I told you that during metaphase or immediately after S phase, the DNA will have two chromatids, right? So very distinctly in metaphase, you can see how there are two distinct chromatids. Let me write it here. This is one chromatid, this is the other chromatid. These two chromatids are held at the center. The region at which they are joined together is called the centromere. Two chromatids belonging to the same chromosome are called sister chromatids. Sister chromatids are identical copies. Okay. So whatever information is there here, whatever is the DNA sequence here, this will have the exact same DNA sequence. They are both identical copies of each other. So they are called sister chromatids. Now if you see, this is the centromere region, the primary constriction about which we've spoken in the chapter Cell, the Unit of Life. On either sides of the centromere, you can see disc-like structures present, which are proteinaceous in nature. It is made up of protein. And those structures are called kinetochores. Okay. So they are called kinetochores. They are disc-like structures that are present on either sides of the centromere. Now these kinetochores can be imagined as double-sided tapes. Okay, wait, I have it right here. Show it to you. So these are double-sided tapes, right? So on one side, it has glue. You can stick it onto something. On the other side, if you remove this wrapper, this side is also glue. So this has glue on both sides. It can stick onto both surfaces, right? The kinetochores are very similar. On one sticky surface, it is attached to the chromosome. On the other side, it is attached to microtubules, which are present in the spindle fiber. Okay, I told you at the end of prophase, spindle fibers formation has started from the aster. So during metaphase, all of these spindle fibers will get attached to the chromosomes by means of their kinetochores. So this sticky kinetochore is bound to the chromosome at the centromere. On the other side, it's bound to microtubules. Okay. Yeah. So now they are positioned at the center of the cell. You can see how the chromosomes are attached to the spindle fibers, specifically at their centromere sites, not at random places, not at the tips. At the centromere itself, it will get attached. This, like I mentioned earlier, is called metaphasic plate or metaphase plate. Now, each chromatid is connected to one side of the pole by means of its kinetochore. The same chromosome's other chromatid is connected to the opposite pole. I want you to notice that it's not like any chromosome is connected only to one side of the pole. Each chromosome is connected to both sides of the pole. One chromatid to that pole, one chromatid to this pole. Okay. Now, next is anaphase. During anaphase, something very beautiful, very organized happens. I'll tell you what it is. You see how the chromosomes are arranged at the center here. Whatever is there information in this segment of DNA, like this chromatid, same thing is here. Now we are going to divide each chromosome such that the subsequently following daughter cell will get one chromatid each. Okay. Now you want the chromosome to split at the centromere and make sure each cell receives one chromatid. Over here, very sequentially, all of the chromosome centromere will split simultaneously. This does not happen in metaphase. I'm showing it to you in this diagram, but this actually happens in anaphase. So all of the centromeres of all of the chromosomes that are aligning at the center, they will split at the same time, simultaneously. Here there are only 4 chromosomes. Imagine in our cells, there will be how many? 46 chromosomes. Every time mitosis is happening, 46 chromosomes will come and align at the center of the cell. All 46 of them at once simultaneously will split the centromere and the two sister chromatids will get separated. Now that the sister chromatids have gotten separated, you no longer call them sister chromatids, you call them sister chromosomes now. Okay, so during anaphase, the chromosomes are arranged at the metaphase plate, splits, not just splits, it splits simultaneously. The two daughter chromatids are now referred to as daughter chromosomes of the future daughter nuclei. They will start getting pulled towards the opposite poles. I'll tell you how this pulling also happens. You know how microtubules can be polymerized and depolymerized, right? Microtubules are made up of tubulin proteins. If you add more and more tubulin proteins, tubulin dimers, it will start getting polymerized, increase in length. If the tubulin proteins are getting removed from one end, like peeling of a banana, it becomes shorter and shorter. So the microtubules shorten in length, and as the microtubules here, the spindle fibers become shorter and shorter, the chromosomes that are attached to the microtubule by means of its kinetochore will also become shorter and shorter. That way it gets pulled towards the opposite poles. When it's getting pulled towards the opposite poles, since they are attached to the chromosome by the kinetochore, which is present adjacent to the centromere, it will appear like the centromere is moving towards the pole. It's in the leading end and the two tails are trailing behind. It's very similar to how if you have like a piece of cloth that I can show it with you. If I'm trying to move this piece of cloth towards one end, let's say I've tied a thread around this. This is the point of attachment and I'm trying to pull it upwards. It's obviously the point where I have attached the thread that will be forward. The other two parts will be trailing behind, right? Very similarly, even over here, the centromere will have the leading edge and the arms trail behind. All right. Now, this is the structure of microtubule. That is what spindle fibers are made up of. They are made up of these tubulin proteins. So here you can see how in a microtubule, if you add more and more tubulin proteins, it increases in length. But when cell division has to happen, it will have a minus side as well, where it will get depolymerized. Once it gets depolymerized, it will become shorter and shorter, and it will carry the chromosomes away from the center. Okay, so that's what happens during shortening of the spindle fibers. Now let's talk about the last phase of karyokinesis in mitosis, which is telophase. Telophase, exactly whatever happened in prophase, everything will be ultified, reversed. So during telophase, which is the final stage of karyokinesis, the chromosomes that were condensed will start becoming decondensed. From the center, they would have reached the poles. So once they have reached the poles, they will start decondensing from being compact chromosomes like this. It will start becoming chromatin fiber. Okay, so it will start decondensing. Yeah. And individual chromosome identity you can no longer see. They will no longer appear like this. Individually, you cannot see which is which DNA, which is which chromosome. They will start appearing like this, where they lose their individual identity. Everything will start appearing like threads. You can't separate, you can't say that this DNA, this chromatin fiber is from chromosome 1, this chromatin fiber is from chromosome 2. You can't say that because they become fully decondensed. Okay. Yeah. And you can see how the nuclear envelope will reform, nucleolus will start reappearing, Golgi, ER, all of them will reform, all of them that were standing in the side, making space, providing space and stage for the chromosomes to get separated. After the chromosomes have been successfully segregated, they will start, reforming around the DNA, because DNA is a very important material. It is the genetic material. It has very important precious information that should not get changed. It should not get mutated. So it needs protection. Once the job of division, separation of chromosomes is done, these nuclear membranes will form around the nucleus. And that's the end of telophase. Now, from one cell which had one nucleus and one centrosome, we've reached to one cell which has two nuclei and two centrosomes. The next job is to split the cytoplasm and make sure they become two entirely different cells, which is exactly what we are going to achieve in cytokinesis. Cytokinesis, like I mentioned, is the division of cytoplasm. The process is quite different for plants and animals. In animals, a furrow forms. Okay, so if this is the cell that has two newly formed nuclei, a ring of actin forms over here. Imagine it in 3D. Imagine this like a oval shaped cell. So a ring of actin forms here, and that will contract. When the ring contracts like this, actin, myosin capable of contraction and all of that, right? So over here, the same actin will form a ring at the center and it will start contracting. When it begins to contract, a furrow will form here. Okay. So this furrow will deepen and deepen and deepen, and similar to how you take out chapati dough to roll out chapatis or rotis from the big fat dough, very similarly a furrow, or it gets pinched such that from the outside, from the periphery to the center, a groove forms or a furrow forms, and eventually the two furrows from opposite ends, they will meet. Like, think about it like a balloon that's filled with water, holy watercolor balloons. So color balloons, whatever balloons. So when you squeeze that from the center, you can see how the two ends will become slightly bulged and the center will become snatched, right? So very similarly here, the center will become snatched. It's like the entire ring is getting contracted. It's not like only two ends. It's like the whole ring at the center, the whole circumference at the center, it gets snatched, and as a result, the two cells will separate. So in animals, this is how it happens. It starts from the periphery and towards the center it will move. But in plants, that's not the case. In plants, a cell wall needs to form. I've told you about how plant cells In plant cells, new cell wall will form. In the previous chapter, which is Cell, the Unit of Life, when I spoke to you about the plant cell wall, I will briefly repeat it again. Here is the newly formed brand new nuclei. So there is a scaffolding that is laid with the help of microtubules and other cytoskeletal elements. So from there, middle lamellae formation will happen. Calcium pectate will start forming. That is called the cell plate. Okay. So from the center to the periphery, the cell wall will start forming, and that is how the two nuclei cytoplasm will get separated. That is how cytokinesis happens in plant cells. It happens by formation of cell plate. Here a furrow forms. With that, we've come to the end of how mitosis happens. Now we are left with how meiosis happens, which is the tougher type of cell division. So let's get started with meiosis. Oh well, I seem to have forgotten about the significance of mitosis. Let's understand that first. There are two very important roles that mitosis plays in our bodies. Firstly, it helps in growth. Like I mentioned at the beginning of the class itself, we all started our life as a single cell, but today we're made up of trillions of cells. How did that happen? Thanks to mitosis, we divided and divided and divided, and we became multicellular. So growth in us happens because of mitosis. And secondly, repair also is very, very important function or significance of mitosis. You know, when we eat hot food, the buccal epithelium kind of gets burnt. Because of the highly acidic conditions of the stomach, the epithelial lining very frequently gets corroded. The skin on our body, these are also very frequently replaced cells. So, or you fall off somewhere and there's a cut in your skin that gets healed, right? All of that happens because of mitosis. If newer cells could not form very well, our buccal lining or the lining of our stomach, all of that would get corroded, right? Because cells are getting replaced very frequently, newer cells are getting added. Wherever damage has happened, their repair is happening. That is why we are capable of existing, surviving the way we do. So, for cellular repair also, mitosis plays a very, very important role. Now, speaking of meiosis, meiosis, like I mentioned earlier, it's also called reductional division. Because over here, when one cell divides to undergo two cells, I mean actually not two cells but four cells will form from one cell. If the parent cell is diploid, all of these cells will be haploid. So if the parent cell has 46 chromosomes, exactly half the number of chromosomes will be present in these cells. When I say it gets reduced, the number of chromosomes get reduced, it does not get randomly reduced. If a parent cell had 46 chromosomes, the daughter cells will not have, one will not have 44, another one will have 43. It's not any random reduction. It is exactly in half. Okay. So that is what happens during meiotic cell division. And in haploid cells, further meiosis cannot take place unless the cell has an even number set of chromosomes. It cannot undergo meiosis. A triploid cell cannot undergo meiosis. A tetraploid cell can undergo meiosis. If it has 4 sets of chromosomes, the daughter cells will get 2 sets, half the number. If the parent cell has 2 sets of chromosomes and is diploid, and then the daughter cells will get a haploid set, 1 set. Okay. Now, every time during meiosis, from one cell, 4 cells only will form. There is no change in that. During mitosis, from one cell you get 2 cells. During meiosis, from one cell you get 4 cells. Now, when does meiosis happen? Meiosis happens during gamete formation or gametogenesis in diploid organisms. I told you the example of honeybee males who are haploid, right? In them, during sperm formation, during gamete formation, meiosis will not take place, mitosis only will take place. If the organism is diploid and sperms or eggs or gametes have to form, only then meiosis will take place. Okay, so in us, during formation of sperm and egg, which are male and female gametes respectively, you can see that meiosis will take place. Now, when meiosis happens, half the number of chromosomes are form in the daughter cells. Why is that essential? That is essential because that helps in maintaining the chromosome number in a population. If you look at humans, all normal humans will have 46 chromosomes only. If it has more or less, that is an abnormal condition. How is it that every human has 46 chromosomes? It's because of this process called meiosis. Because of meiosis, there is a haploid phase in our life cycle, which is the formation of gametes. Again, when the gametes fuse and fertilization happens, the diploid number will be restored. If only meiosis did not take place and mitosis only took place during gamete formation also, we having 46 chromosomes, our sperms and eggs would have 46 chromosomes. If one person's sperm has 46 chromosomes, fuses with an egg that also has 46 chromosomes, in the next generation we would get zygotes that are having 92 chromosomes. What if that person, that baby grows up and decides to reproduce and mitosis happened during gamete formation, that would produce sperms or eggs that had 92 chromosomes. And it wouldn't be impossible for an entire population of humans, all the individuals, normal human individuals, to have the same number of chromosomes. Different individuals would have different number of chromosomes. That is not usually how nature works, right? In most cases, if you look at individuals belonging to a species, they will more or less have the same number of chromosomes with a very few exceptions. The reason why the chromosome number is maintained in a population is because of this process called meiosis. So, in meiosis, because reduction has to happen, reduction of the number of chromosomes have to happen, meiosis will happen in 2 cycles, okay. Similar to how mitosis had prophase, metaphase, anaphase, telophase, meiosis also will have prophase, metaphase, anaphase, telophase, but not once, twice. So, in meiosis, there are 2 sequential cycles which we simply call meiosis 1 and meiosis 2. But in between meiosis 1 and meiosis 2, there is no DNA replication that happens again. From meiosis 1, common to both meiosis 1 and meiosis 2, DNA replication happens only one in the S phase of the cell cycle. Okay. So, after a single cycle of DNA replication, you can see meiosis happening. Okay. Meiosis not only helps in reducing the chromosome number, it also brings about variation. How exactly it brings about variation? As we understand the process, you will get to know. Like I already mentioned, in meiosis, from one diploid cell, you get 4 haploid cells. So, you have interphase G1, S, G2, then meiosis is happening. In meiosis, again you have meiosis 1, meiosis 2. In meiosis 1, again you have karyokinesis, which includes prophase, metaphase, anaphase, telophase, prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis of prophase 1, it may or may not happen. Following that, you have karyokinesis of meiosis 2, where you have prophase 2, metaphase 2, anaphase 2, telophase 2. Following that, you have cytokinesis again. So, it's a very long process. I spelled it wrong, cytokinesis. Alright. So, it may be long, but it's super interesting. It's one of the coolest processes to ever exist. Okay. So, let's learn how it happens. I'll give you an overview of what happens during meiosis in our cells, and then we will learn about what exactly happens in every phase. So, if you think of this as the cell cycle, here, so that there's sufficient space to write things out. So, here you have the interphase, which occupies the most space, most part of the cell cycle. Let's just call this the interphase. In the interphase, you have G1, you have S, and you have G2. Over here, you have meiosis 1 and you have meiosis 2. Okay. So, let's start off with a cell that has just been formed. It has entered the G1 phase. So, the cell is going to be very small in G1 phase. It will have, let me just show the DNA content also. Let's say it has 1, 2, 3, 4 chromosomes. Not like chromosome, it will exist in the form of chromatin. Just so we understand what happens, I am going to show it a little bit as chromosomes itself. So, as it moves through the G1 phase, you can see how the cell will increase in size. Okay, but DNA content will not change. It will still have chromosomes, 4 in number. Of course, I am drawing 4 because don't expect me to draw 46. it'll have the same 4 number of chromosomes, each chromosome having 1 chromatid. Okay, so it's just increased in size. Here also you have 4 chromosomes, 1 chromatid each. Now we call this the 2C condition. Over here the cell has grown in size, no change with respect to DNA. So here also you have 4 chromosomes, 1 chromatid each. So this is also in 2C condition itself. Next what happens is S phase. So as the cell proceeds through S phase, at the end of S phase, the cell would have grown a little bit bigger in size. It would have replicated the DNA also, and at this point you have 4 chromosomes, but each chromosome has 2 chromatids each. The number of chromosomes remains the same, but instead of 1 chromatid each, it has 2 chromatids. So you have 4 chromosomes. Each chromosome has 2 chromatids. Now, we say that it is in 4C condition. Next, what happens through the course of G2 phase? Nothing changes. The cell may increase in size slightly, okay? But even in G2 phase, you can see how the chromosome number does not change. The DNA content does not change. Whatever was there in the S phase, the same thing will be even over here. I'm not showing nucleus and organelles because our focus right here is what's happening with the DNA. The nucleus will be there, other organelles will be there. This will not exist as chromosomes, it will exist as chromatin fibers, but if it's like that, you will not be able to understand. That's why I'm saying chromosomes, okay? So here at this point, you still have 4 chromosomes. And how many chromatids each? 2 chromatids each. Right, ladies and gentlemen, boys and girls, are you sleeping? Are you still listening? If you're still watching this, let me know in the chat. Okay, so this is still, 4C condition. Okay, nothing has changed from here to here. Now what happens in M1 is interesting. As the cell proceeds through the first meiotic division, 2 very special things happen. Firstly, one cell will divide and give rise to 2 cells. Okay, this is happening in M1 phase. After M1 phase is telophase, you can see this. 2 cells will form from this one cell. And the chromosome number would have changed here already. From 4 chromosomes, each cell would have 2 chromosomes here. This is one chromosome, this is the other chromosome. So we all are diploid organisms. We said we get one set of chromosomes from the mother through her egg, we get the other set of chromosomes through our dads through the sperm, right? So here assume that the chromosomes that I'm showing in cyan color are paternal chromosomes, and the pink ones are from the mother maternal chromosomes. So after the first meiotic division, it so happens that each cell receives one copy of the maternal chromosome, one copy no, that's not what I meant to say. Okay, so before that, let me tell you this. Now, in a cell, you will have 22 pairs of chromosomes. In this case, we have 2 pairs of chromosomes. One chromosome, 2 chromosomes are from the father, 2 chromosomes are from the mother. Okay. I know that these 2 chromosomes are one pair and these 2 chromosomes form the other pair. How do I know that this chromosome is the same as this chromosome? These both are in the first pair, these both are in the second pair. It depends on the kind of genes it carries. If this chromosome at this position has information for hair color and this chromosome at the exact same position has information for hair color, then these two are said to be homologous chromosomes. They have similar information at the same place for similar character. Okay, so these are called homologous chromosomes. These two chromosomes at that location, they probably have information for something else. They probably have information for, let's say, hair texture. Okay, so these two have information for the same characteristic at this location. That's why these both are homologous. This has hair color information, this has hair texture information, so they are not the same pair. They are not this homologous. Homo means similar, right? So both of them have similar genes for similar characters. They are homologous. These both are homologous. So when meiosis I happens, homologous chromosomes will get separated. Okay. Now when homologous chromosomes get separated, I have shown you how each cell will receive two chromosomes. Initially, there were 4 chromosomes in the parent cell. Each cell received 2 chromosomes. As meiosis I proceeds, something beautiful happens such that these chromosomes will not be either entirely maternal or entirely paternal. A process called crossing over will take place such that each chromosome becomes a mixture of maternal and paternal genes. So it will no longer exist as strictly paternal or strictly maternal. Crossing over will happen in such a way that all the chromosomes will have combination of maternal and paternal parts within it. This happens during M1 phase, and this is very, very important. This is one of the hallmark things that happen during meiosis. Now, if you look at the cells post meiosis I, you can see how each cell has 2 chromosomes, 2 chromatids each. Since the DNA content of this is similar to the DNA content of this is also called 2C condition. But you might argue, ma'am, but there are 2 chromatids in a chromosome. 2C and 4C represent the DNA content and not whether the chromosome has 2 chromatids or 1 chromatid. Look at the DNA here, look at the DNA here. Here you had 1 chapati, you had split it into 4 parts. Content is the same. Here you have 1 chapati that you've split into 2 parts. Content is the same. Here you had how much ever amount of DNA you had, same amount of DNA you will have here also. So both of these have equal amount of DNA, that is 2C condition. If you notice here, the number of chromosomes have already gotten reduced. What was 4 chromosomes per cell has now become 2 chromosomes per cell. I told you how meiosis is reductional division. So the reduction in chromosome number has already happened in meiosis I itself. Following this, mitosis will happen. When mitosis happens, this will give rise to 2 cells and this will give rise to 2 cells. Okay. And meiosis II will be exactly like mitosis in the fact that the centromere will split, okay, and the chromatids will get separated out. So at the end of meiosis II, which is like mitosis, you will see how each cell will end up having 2 chromosomes, which are mixture of both the parents. This is how it brings about variation in a population. This is how it ensures that newer genetic combinations are created in a population. So at the end of meiosis, you can see how each cell will have 2 chromosomes and 1 chromatid each. Now that it has half the DNA content that this cell had, we can say that it is in 1C condition. What was 2C became 4C, became 2C, eventually 1C. So these are the final 4 haploid cells that will form from one diploid cell at the end of meiosis. This may have been a little difficult for you to comprehend. So I will explain to you what exactly happens during crossing over as we move on. This is just an overview. If you haven't still understood what happens during meiosis, don't worry, we will discuss that in detail. Okay. Now, speaking of meiosis I, we'll start with prophase I. In prophase I, you further have 5 substages or subphases, which are leptotene, zygotene, pachytene, diplotene, and diakinesis. So, unlike prophase of mitosis, prophase I is typically longer because it is during prophase I that genetic rearrangement happens, crossing over happens, and newer combinations are formed. Okay, so let's see. Okay. We'll learn about what exactly happens in each one of these phases, starting with leptotene. Leptotene is the first phase of prophase I, very similar to what happens in the prophase of mitosis. DNA will start getting a little bit condensed here. So the chromosomes will start becoming gradually visible under the light microscope, and the compaction of chromosomes becoming more and more condensed and compact will continue throughout leptotene. The next phase, zygotene, is a very, very important substage of prophase I. It is a second phase. Over here, something very interesting happens which we will need to understand. Okay, so let's say a person has to produce gametes. A person is producing gametes. That's when meiosis will happen. For example, when that person is producing gametes, let's assume that person had parents. That person had parents whose mother, this is the father, let's say, and this is the mother. His parents had, let's say, his dad, this is his dad, and this is his mom, of course. And the dad had red hair that was straight. Okay. And his mother had blonde hair that is curly. So this guy has received one chromosome from the dad and one chromosome from the mother. Both of these are homologous chromosomes that I'm talking about. So let's say at this position on the chromosomes, there is information for hair color. At this position on the chromosome, there is information for hair texture. This person has hair color information here as red and curly. The woman on the other side in her chromosomes will have information for blonde hair that is straight or what? Curly. So this guy had straight, no? Sorry, I mixed it up. This woman is curly. This guy has straight hair. Okay. So this person has both of these chromosomes inside his cells. When he produces gametes, only one of these will go into the gametes, enter into the gametes. If only one of the parents' information is getting passed on to the next generation, the offspring will be more similar to one of the grandparents than the other. Excuse me. In order to make sure that the offspring gets more variations of genetic material, gets a mixture of both grandfather and grandmother's genes, when this guy is producing gametes, crossing over will happen between these two chromosomes, as a result of which newer genetic combinations will arise. How exactly that happens, I'll tell you. So this is how it is in this guy's cell during interphase. Okay, so in interphase, this is how it will look. Let's say S phase happens. After S phase, you will notice how each chromosome will make an extra copy of its chromatid. So after S phase, this is going to be the case. But during S phase, if here red was there, here information for straight was there, here also same thing will get copied because the exact same replica is happening. Here if there was information for blonde hair that is curly, same information will get copied. So these two chromatids here And these two chromatids here have identical information, and they are called sister chromatids. Okay. These two have information at the same location for the same character. So these two chromosomes are called homologous chromosomes. Alright. Now, the two chromatids of two different chromosomes belonging to the same homologous pair, they are called non-sister chromatids. Okay. So this chromatid from here and this chromatid from here, they are called non-sister chromatids. But it has to be a part of the same homologous pair of chromosomes. It can't be this is one, this chromosome belongs to the first pair, another chromosome belongs to the third pair. They are not homologous. In the homologous chromosomes itself, this and this of two different chromosomes belonging to the same pair, they are called non-sister chromatids. Now, if both these guys had black hair, it's possible that this gene in both can be the same. Now, for example, I've taken blonde and red hair. So, the information that is present here is different. It's not necessary that in non-sister chromatids, every time different information only has to be present. Both my parents have black hair, both my parents have wavy hair. So, in my chromosomes, it will be the same. I am trying to say that it need not necessarily be different each time. For it to be non-sister chromatids, it does not mean it has to carry different information. It could carry the same information also. Why we call it non-sister is because it is not part of the same chromosome. It is not always identical. It can be different also, but it need not necessarily be different. Okay, so these are your non-sister chromatids. This is your homologous chromosomal pair. Okay, so what happens during packaging is the chromosomes, zygotine, we were in zygotine only, no? The homologous chromosomes will pair like this. If this is one maternal chromosome, on top of this, there will be like a synaptonemal complex, a sort of glue that forms, and that particular glue will keep the two chromatids, the two chromosomes attached to each other. I don't know how to show it in 3D. Assume that this is one chromosome, this is the other chromosome. It's attached to each other like this. Okay. So what is gluing them together is called synaptonemal complex, and this complex of two homologous chromosomes sticking to each other is what we call synapsis, that is chromosomal pairing. Random chromosomes cannot pair, homologous chromosomes only will pair. Now that you have two chromosomes together forming a complex, you can call it a bivalent. Because it has 1, 2, 3, 4 chromatids, you also call it a tetrad. Bi means 2, tetra means 4. 4 chromatids are there and 2 chromosomes are there. Okay, so the bivalent will start forming in zygotine. Remember that throughout the course of prophase 1, leptotene, zygotine, pachytene, diplotene, diakinesis, DNA condensation is continuously happening. It is a continuous process. From one phase to the next phase, it will be more and more condensed. So condensation is still proceeding over here. Okay, it started in leptotene, it's proceeding here. What happens in pachytene is the bivalents or the tetrads will start appearing more distinct and clear because obviously it's getting more and more condensed with passing time as it moves through different phases, right? So over here specifically, you can see a process called crossing over taking place. For example, if this was one chromosome, daddy's chromosome, he had information for red hair in this position. On top of this, lying right now is mother's chromosome. She has information for, what is it? Blonde hair at this position. Okay. This guy has information for curly hair at this position. And the woman has he has it for straight hair, and the woman has information for curly hair at the exact same position. Imagine it in 3D, okay? Now what happens is, in specific segments of DNA, chromosomal exchange will happen. in between non-sister chromatids. These two are non-sister chromatids, these two are non-sister chromatids. There's no point when exchange of material happens between sister chromatids because they are identical. Red hair information will go to straight hair will go to straight hair. Newer combination will not form. But if in between non-sister chromatids crossing over happens, you can get red hair that is curly, blonde hair that is straight, different combinations can arise. Okay. So now what happens is crossing over will happen. When crossing over happens, an enzyme called recombinase mediates that process. It's not like the entire segment will get exchanged. That will also serve no purpose. Newer recombinations will not form. Newer recombinants will not form. So at specific segments, crossing over between the genetic DNA molecules or the chromosomal segments will happen. So when that happens, see, in this chromosome, let's say at this segment, crossing over has happened. Because crossing over has happened at that segment, now you have the female's DNA at this point. And in the same chromosome, let's say crossing over has happened at this point also. Okay, so this guy's chromosome, this chromatid is changed over here. Another chromatid is same over here, but here at the bottom, it has a segment from exchanged from the maternal chromosome. Now this chromosome became a mixture of male maternal and paternal chromosome DNA. This also became a mixture of maternal and paternal DNA. What happened to the other one? That also got some of the segments exchanged. Okay. So here you can see this has this portion, this has this portion. Now if you look at these chromosomes, they are not yet separated, they are present right on top of each other, but they have crossed over. I am just showing it to you here in this way, but they are still together, not separated. In this chromatid, you can see that there is information for blonde. This is red information, red hair information. This is blonde hair information. This is information for red hair color. Over here, there is information for straight hair because it is paternal from the father. Here there is information for curly hair. Here there is information for curly hair. And here there is information for straight hair. What was the initial combination we had? We had red hair that was straight, blonde hair that was curly. Because of this process of recombination, look at what has happened. Blonde hair that is straight, red hair that is curly, we've gotten two new combinations, right? So that is possible only because of crossing over. Newer combinations will appear in the offspring because crossing over happened. Okay. So if crossing over did not happen at this place, at least some chromatids would have retained the parental combinations. Again, it depends from region to region wherever crossing over has happened. It will happen somewhat randomly itself. Okay. So I hope this has given you an idea how newer combinations can arise. Initially, red was with straight, curly was blonde. Now you have red curly and straight blonde. So that's how even in us, we have a mixture of characters, some like mother, some like aunt, some like grandfather. That's how it happens. Okay. So this process called crossing over is a characteristic feature of the Pachytene stage. Whichever locations crossing over happens, there nodules, sort of oval regions or swelling sort of things will happen. That is called recombination nodules. Specifically at those regions, you can see crossing over happening between non-sister chromatids of homologous chromosomes, never between sister chromatids, because even if it happens, it's pointless because both of them are identical. Okay, so it's important for you to remember this happens between non-sister chromatids. Now what happens during diplotin is both of these chromosomes will start getting separated from each other. The synaptonemal complex that was acting like a glue holding the two homologous chromosomes together will start dissolving. That is called dissolution of the synaptonemal complex. So that will begin in diplotin, and except for regions where crossing over has happened, in all other parts it will separate. So wherever crossing over has happened, over there you can see X-shaped structures that are called chiasmata. You may have heard how in humans, especially in females, when oogenesis happens, it begins before the baby is even born. Right when I was in my mother's womb, I had started producing, oogenesis had begun in my ovaries. But that meiotic division will not continue. It will be arrested at one stage in prophase 1, typically in diplotin, not just in our oocytes, in the oocytes of many other vertebrates as well. You can see how the cell cycle will be arrested at diplotin and it will stay that way for many, many years. In us, it was arrested in diplotin even before we were born. Up until we attain puberty, it will be in diplotin stage itself. Okay, so that's an important point. Ultimately, what happens? Diakinesis will happen. Diakinesis is like the final stage of prophase 1. So throughout the course of prophase 1's many subphases, you need to understand the DNA is becoming shorter and shorter, more compactly forming into chromosomes. So continuously, even after diplotin also, it will start getting condensed. I told you how these X-shaped structures called chiasmata are keeping the two homologous chromosomes together. So basically, when DNA gets condensed, it will appear like these chiasmata are moving towards the tip. As the chromosomes become shorter, it will appear like this is moving towards the tip. We call that terminalization of chiasmata. Since diakinesis is the last phase of prophase 1, over here the DNA will be fully condensed, nuclear envelope disappears, nucleolus disappears, ER and Golgi disappear, and it will transition into metaphase. If it's an animal cell, like I mentioned earlier, the centrosomes will move towards opposite poles, all of that will happen. Mitotic spindle will assemble. Yeah, so that's basically what happens during diakinesis. Now moving on to what happens during metaphase 1. This is what happened during prophase 1. Now you can no longer say that after prophase 1, you can no longer say this is maternal chromosome, this is paternal chromosome, because both of them would have mixed up. Each chromosome will have certain segments of father's genes. Each chromosome will have certain segments of mother's gene as well. So it would have all become a mixture. That is what is represented here. It is shown in different colors. I hope you can see this in your textbooks. So during metaphase, the chromosomes will come and align at the center, very similar to what happens during mitosis. But the difference here is it is arranged not in one line, but it is arranged in two lines. Here you have two metaphase plates. Okay. So both of these belong to one homologous pair. These two belong to one homologous pair. They are arranged like this. Over here, you can see how the centromeres will pull, the spindle fiber will pull the centromeres or the chromosomes towards the opposite pole. During anaphase, you don't see centromere splitting here. In meiosis, the kinetochores are arranged such that it's present only on one side. Okay. So from one side only it will get attached to the spindle fibers and the entire chromosome will be pulled apart. So from having 4 new chromosomes in this cell, you will end up having 2 chromosomes each. Over here, chromatids are not separating. What is separating is homologous chromosome. These both were part of one pair. Now they both are getting separated. These both were a part of one pair. They are getting separated. So homologous pairs of chromosomes are being pulled apart and they are getting separated. Why can't these two go into one cell, these two go into one cell, you might wonder. This cell has information for the same thing, this cell has information for the same thing. So you will get double dose of information for one thing and no information for this. What if this had essential genes like ATP synthase producing genes? Then this cell will get incomplete information, this cell will get incomplete information, right? So that's why, to avoid that, the homologous chromosomes will get separated during anaphase 1. And in telophase 1, nuclear envelope will form. From one cell you get 2 cells. This is called a dyad of cells. So in metaphase, cells will align at the equatorial plate, microtubules attach at the kinetochore of the chromosomes. At anaphase, what is separating? Not the chromatids, it is the homologous chromosomes that separate. The 2 chromatids will still be associated at the centromere. They will not, the chromatids will still be associated at the centromere and they will not yet split. Next, in telophase, nuclear membrane nucleolus reappears. Since you have 2 cells right now, you have a dyad of cells. So after telophase, little bit of decondensation of chromosomes will happen, but it's not as much as it is in interphase. After telophase 1, you have cytokinesis, and after cytokinesis, you have a very brief phase that is called interkinesis. Okay. So during interkinesis, it's not like interphase where DNA replication will happen. This is just a very brief period, resting period for the cell. It's very short-lived, very brief. Over here, replication of DNA will not happen. This is just a very small phase between meiosis I and meiosis II. After interkinesis, you have meiosis II, you have prophase II, metaphase II, anaphase II, telophase II, cytokinesis II. So what happens during prophase 2, metaphase 2, anaphase 2, telophase 2 is exactly what happened during mitosis. Meiosis II resembles a normal mitosis. So during prophase, DNA condensation happens, spindle fiber starts to form, nuclear envelope disappears, all of that will happen. During metaphase, the chromosomes will align at the center of the cell. During anaphase, the centromere splits at this point and pulls the sister chromatids towards the opposite poles. They are no longer called sister chromatids, they are called sister chromosomes. And ultimately, towards the end of prophase 2, telophase 2, you can see how the nuclear membrane reappears, how the DNA will start to decondense, all other organelles will start to take shape within the cell. And ultimately, following cytokinesis, from one cell, we end up getting two cells. So we started off with one diploid cell, we end up getting 4 haploid cells in the end. That is the story of meiosis. Meiosis has two very important significance. Firstly, it helps in maintaining the chromosome number, conserving the chromosome number in a particular species. Secondly, it increases genetic variability. How does it do so? Because of the process of crossing over. If not for crossing over, our children would get either completely one set of my father's genetic information or one set of my mother's genetic information. Because of crossing over, each chromosome becomes a mixture of maternal and paternal genes. So there is newer recombinants formed. That is why when two children are born to the same parents, they are siblings, but still they don't look identical to each other. They are all the time newer combinations in their characters unless they are identical twins. So that's about the significance of meiosis. And with that, we are ending our lecture for today. I hope any doubts that you had in this chapter have been clarified. If you have any more doubts, you may please feel free to ask them away in the comment section. I will respond to them as soon as I can. So with that, I will sign off from today's session. I will meet you again in the next class next week. Take care of yourselves, stay healthy, stay happy, and thank you.